like to invite our presenters to come up for our panel discussion. I'd also like to invite 3 additional individuals. We may need some more chairs, I'm not sure. But Richard Compton from Nanopore, Joshua Trotta from Thermo Fisher, and also Christian Hanisch from Illumina. If you guys would join us up here, we'd greatly appreciate it. So now is the time for your questions. If you could make your way to the microphone. Are we okay on chairs?
We're waiting for the music to start.
Sorry.
As long as I don't play.
I think we're good. Okay, and we have some microphones up here on the table as well. So our first question, please.
Hi, I'm Eric Hout from Virginia. How do you envision doing the clinical trials to evaluate the amplicon-based sequencing approach against presumably a phenotypic method as the gold standard? Like a couple of questions that come up are, you're going to have heteroresistance to possibly deal with, have to have some number, you know, that you're using to compare it to. And then if the data looks like what Matteo shows, you know, from these large series for PZA and moxifloxacin, which are probably, you know, the 2 really important drugs to have new methods for, if you get 60s to 70% sensitivity for resistance, which is what Sanger, you know, and other approaches yielded, would that even get approved? How could that get approved by a body? So those 2 questions.
Yeah, and there's actually quite a bit in there, but I think I'll just try to hit on a couple of them, and remind me if I'm not hitting. So, you know, what are we going to compare to? How are we going to do those comparisons, and then specifically with PZA and Moxi? So, you know, right now we've been doing a number of validation and verification studies in preparation to get ready for doing that type of a clinical trial. So we've been comparing to the other gold standards that are out there. Obviously the gold standard of phenotype testing for midget, but then we're comparing to other molecular tests. So comparing to pyrosequencing or Sanger sequencing. And we have papers either out on that or they're being reviewed right now. And just to let it out of the bag, it performs very well, as we expected it would. And then, sorry, I've had 3 Benadryls today. I don't know if it's my allergies. So now everything's starting to run together. And so, you know, we actually have a couple of partners that want to be kind of first in line to help us with doing clinical trial, but focus on just a couple of the markers, which I think actually makes a lot of sense rather than trying to push it all through at the same time. And so with PZA, we actually have limited comparison there, but we see essentially 100% sensitivity in picking up PZA resistance. We actually, you know, we look at the entirety of the whole PNCA gene, and so you know, we can look down to fairly low levels, and that gets back to your heteroresistance question. The TPP that Tim talked about really says what we want to aim for is at least 10%, because 10% is about the lower limit at all possible by Sanger, which is probably more like 25%, or pyrosequencing as well. So we know that we can identify it at 10%. We can identify down to 5%, but what we'll probably end up having to do is just classify everything in between 5% and 10% is going to be heteroresistance identified, but the relation to phenotypic resistance is unknown.
Barry?
Yeah, couple of things there. You hit on a very important point. If you're basically pioneering a method that doesn't have a good standard, how do you make a good standard? So some of it goes back to Sanger, but then what are you doing Sanger on? If you're doing Sanger on culture is already different than doing it directly from sputum. So basically we're doing 4 or 5 different ways to try and create a 360-degree view of not only what we get, but why we get what we get. So absolutely there's going to be discordance with phenotype. I think what Matteo was talking about, the lower percentage sensitivity with moxifloxacin at 2.0, We all know why that is. 2.0 is too high. That is not the ECOF for moxifloxacin. And so we're basically going to have to compare things to ECOF relative to critical concentrations. That's a whole big conversation that's just about to happen at WHO. A lot of those things will probably get redesigned or reaffirmed after that meeting coming up in April. So I think there's a whole bunch of ways that we're going to have to deal with it. So you're absolutely right. It's not going to be like a regular phenotypic study where you say, oh, we'll do the molecular test and one phenotype or a couple cultures. It's going to need this sort of 360-degree view to understand why we get what we get.
So I have a question for Dave. So in your targeted sequencing, you're only looking at the hotspots. Are you looking at RDR or 315 for CAT-G, or are you going to the whole gene like with PINK-A?
Yeah, so it really depends on the target. So yeah, we're looking at the RDR, the QRDR. In PZA, we're looking at the entirety of the gene.
Okay, so you're taking advantage of by going to direct sputum, which is great, and you're looking at heteroresistance, but you're missing other mutations in rifampin, and you're only looking at one codon in INH. So it seems like you're putting your effort, you're losing so much. So I would rather look at the rest of the gene than worry about heteroresistance, because I think you're missing a big part of resistance that we know there's other mutations in CAT-G, and we know there's other mutations in, so why don't you just do your targeted, but just extend it and add, the rest of the gene so you cover more. And I think your heteroresistance is great, but I think it's a minor point, whereas you're going to get more for your buck if you look at the whole genes, much like you're doing with PINK-A. So yeah. I think you're doing good on one end, and you're sort of cheating yourself on the other end.
No, I think it's a really good point, and one that we're learning. So on a number of these, we are expanding to go out larger than the target regions. We certainly we're going for most bang for our buck. So looking what's going to give us 90 to 98% of resistance when dealing with MDR or XDR. That's where we focused on. And there are going to be lots and lots of mutations outside of that make up a very small percentage, though, of the overall cases. So they're important, and we can easily expand by manipulating the primer set, adding in new primers to be able to capture that. And that's a fairly simple thing. And every time that we identify an area that we really want to capture, we can drop another one. And it takes a bit of validation that it's not going to mess with the other amplicons. Yeah, but so far it's worked pretty well.
Yeah, and I think it would improve your overall assay. And you're going to have data from FIND as well as from the databases to really do this. I mean, that's what the databases are for, to say, look, you're missing something, why don't you just add it to it?
Yeah, it's a really good point.
Looks like everybody's picking on you, but for the next question, maybe somebody else can answer, but they're all falling into the same category.
I'm the one on drugs here.
So it looks like it. So it looks like the notion of pre-resistance, which to me is a new word which I love, and I'll explain to you why. It's, if I understood it right, it's a subpopulation in a heterogeneous population which eventually amplifies. You can capture easily with existing drugs, mutations, because the mutations are known, right?
That's correct.
But for new drugs like bedaquiline, where we're trying to understand mechanisms of resistance, we're now in a situation where we have this resistance popping up like efflux pump mutations. We don't really know if they're real, if they're not real, not enough evidence. So if we focus all the attention, as proposed, I think, by Matteo, to move completely through next-gen sequencing, chances are that we may miss other mutations that really amplifies it. So these new mutations, which probably I should be calling them pre-resistance to bedaquiline, for example, I think it's too early to shift completely for new drugs, for example, to new gen sequencing, and whole genome sequencing may actually add value in those cases.
I think that's a really good point, and Tim pointed out too that certainly FIND's not trying to find the one assay that's going to cover everything, but we see just an enormous opportunity with focused or targeted sequencing that allows us to capture a whole lot quickly, relatively inexpensively, while you're doing additional tests that may take the weeks or months or more.
No, go ahead.
So I'm the guy from the Nanopore team who's here. We love to work with you, certainly getting some data, longitudinal data, that we can take a look at the impact of long reads. Because although the point that you were making about whole genomes versus amplicons is reasonable, I think current technology, whole genome sequencing, will still only map around 80% of the genome. I think we're seeing regularly, 12 to 20,000 base pair reads coming off, quite simple prep, the ability to do low-cost whole genome sequencing now. And we'd love to start to map some of those black holes with you and see whether we can start to better understand resistance that way.
Yeah, in addition, the sequences that have been generated from strains are now analyzed for possible mutation, possible SNPs that may correlate to the resistance to the new drugs. And then there is also the analysis done on the protein, on the structure of the protein, that to try to look for algorithms that may predict to amino acid substitution a true effect on the sensitivity of the strain. So the data, there are a lot of whole genome sequencing data that can be analyzed to predict the pre-existing resistance to new drugs and to interpret the one that comes.
Just a clarification, Kone. There is no intention from our side to drop phenotypic testing for the new drugs in surveillance. I was just talking about rifampicin INH, quinolones PZA, and injectables for surveillance. Of course, the new drugs, bedaquiline, delamanid, will have to be done with phenotypic testing.
One last question for probably Daniela. I was just wondering if you have a skip well issue when we did your validation with 30 strain study, when you compare frozen versus dry. Was skip well an issue?
We didn't compare the frozens versus the dry.
Okay, sorry.
Because we compare the result. We use the results of the frozen plates that were published as the gold standard for the Thermo Fisher plate, because those were the only results available on H37RV done with a method that was the closest available and published. And there was a very good reproducibility, with the exception that some sites that were performing well on a lot of testing, then they had a couple of tests that were completely off. So that made clear that one site was completely off, so we had to remove it from the analysis. But in some other sites, there were a couple of plates that couldn't be interpreted together with the other. So that will need to go back to the data set and really look at what happened at different sites and day by day. But that was the reference, and the agreement was pretty good.
Thank you.
Hi, sorry about my voice. This is Maha from Harvard. I have actually two questions. Well, the first is, I'm wondering why it seems that there's a lot of investment in targeted sequencing, which won't be point of care. It'll be kind of in a reference laboratory, as opposed to kind of targeted arrays that could potentially be point of care. So that's one question. And the second question is for Richard Compton from Nanopore. I'm wondering how, and so you sort of, I think, alluded to this in answer to the prior question, is how close are we to getting a nanopore type technology to sequence directly from sputum a whole genome, a whole TB genome? I just saw a paper came out from Oxford actually that showed that in those samples, in sputum samples that you could extract DNA from, they got very good sensitivity and specificity for a whole genome sequencing for diagnosing resistance. So I'm wondering if you can also comment on that. Thank you.
Yeah, an excellent point, because we've had to decide very early on where we were going to put our efforts, whole genome or next-gen targeted versus other molecular approaches. And the answer is we didn't. We didn't decide on any one thing. What I was talking about today was one approach. That's basically because it's a complicated set of answers. One is there's going to need to be centralized approaches for this, because most of the drugs that are currently available, for example, the short course, can't be used at point of care. They're injectable drugs. They have to be used higher up the chain. Therefore, having a diagnostic that's way down at the point of care doesn't necessarily help you. But we are investing in those too. So the point is we're investing in point of care to go after those sort of array kind of approaches, a lot of the large industry approaches, Hain, Abbott, Roche, BD, they're all going after those kinds of approaches. And so absolutely we're investing in those as well. But we wanted something that was more comprehensive, that would give us more what we could do without any sequencing, without any culture or anything in the way. And NGS targeted was the best balance that we could do. Would we like to do whole genome and have bigger targets? Absolutely. But I'm also being told that it has to cost less than this and can't take longer than that. And so it's a really interesting thing is, it's actually really important for me to come here and say this, because this has been in my head for so long now, and hear people say, well, why the hell aren't you doing a bigger target? And I'm like, why am I not doing, oh, I remember, because it costs twice as much. And I had to be told that it can't be that cost. So it's, what you're seeing is the best of the world that we could put in place by 2019. And we're basically is because the clamor for NGS right now that is usable today is so high that if we don't do something, they're just going to do whatever the hell they want. And so we're trying to do something that is not the best thing, but it's the best thing we can do quickly with the right price, et cetera. But you're absolutely, everything has to be done, and we're trying to chase it all at the same time, including Nanopore.
Yeah, thank you for the question. And a bit of an advert. Thursday morning, my talk will be really going through the work that's been done in conjunction with us at Oxford. And partly I think that I feel like the troublemaker here, because things have been developing so quickly over the last 6 months at Nanopore. When you look back, even during that time, the device was producing around 100 to 200 meg per run. So it wasn't really ready for prime time. We're now seeing 30 gigabases of high quality data per run. So Zam and Louise's paper from Oxford, 400 meg is enough to get you the coverage you need to make very accurate, sensitive calls. So the ability to then load 40 or 50 samples, index them, on a $500 flow cell is now possible. So I think hitting some of the price points that you guys are aiming at is certainly possible. 8 hours, the guys in Oxford did a great job on their own. We're now in helping them. And again, part of my talk will be running through how, there are a lot of levers across that workflow that we're currently, pulling with them. And we hope to get a very simple, portable protocol that can be used in low and middle income countries that takes the workflow down to 2.5 to 3 hours direct from sputum to detection, then on to resistance profiling. I think the other point, it's a really exciting time to be involved. The other area is uncovering those black holes. So if people are interested in what longer reads can do as far as coverage of those hyper-variable regions that potentially are involved in resistance, we'd love to start working with you on that too.
One follow-up question is, you don't really need amplification, right, for the mini
I think
line,
it depends. Certainly from what we're seeing, the number of just the quantity of TB, of active TB, out in the developing world when samples are coming in, we probably don't need to do enrichment. No, but we are working on very fast enrichment protocols right now, along with Oxford. Again, if people are interested, certainly in getting hold of those protocols and deploying them. Daniela, we've spoken about your lab getting hold of them. Jen Gardy was over last week. I was chatting to her, and British Columbia will do some work on it. But really, the more the merrier, the more help that we get right now in understanding what we can be doing and how we can be using the technology. The point he pulls a DNA sequencer out of his pocket, you can just see an idea of the portability here. We're keen. It's certainly the one area, TB, that Nanopore itself is investing quite a lot of time on our own, along with Oxford, on driving the platform forward to see what we can do. But we really need your help. If we can't just create an assay and expect it to be picked up and used. I think we need a lot of guidance and support in validation, education, training, and just the practicalities of getting the technology adopted in the field. Sorry, taking over.
And sorry, one last question from me. And what is the accuracy right now?
So so this has been moving quickly. We've had a couple of recent releases. We have a 9.5 chemistry release, which is in the works, which is coming out. In-house, we're seeing 97% raw read accuracy using 1D squared protocols, which I won't bore everyone, but we're getting to that. So as far as consensus accuracy, if you're doing 100-fold coverage, we're seeing 99.998 are the highest consensus accuracy figures that I've seen. There's also tricks that we can do in relation to tuning the base calling algorithm to overfit for TB. Or, again, looking at the SNP calls that we're making and looking at the accuracy of those and potentially deprecating one or two of those calls, still giving you the accuracy that you need to make a proper call on resistance. But again, there's still a bit of work for us to do, I think. But again, as many of you who are interested in getting your hands on the device and getting started, we'll do our best to support.
Thank you, very exciting.
Okay.
Marco?
Yeah, Marco Schito from the Critical Path Institute. I think it was a great panel. It was really nice to see sort of what we have currently, where the vision as well of where we can go, and from a variety of different perspectives. One thing I actually wanted to get is Joshua from, there you go, yeah. So I'm going to ask you a question, Joshua. And also Christian, so you've been quietly sitting in the corner, listening to all of this. I just wanted to get, for initial gut reaction, anything that has been said or any particular topics that you would like to touch on from Thermo Fisher side, and then also from Illumina. And I have a few other questions, but I just wanted to give you guys a chance to say something.
Thanks, Marco. Thanks for including us, man, and then making sure we're not too quiet down here. So, yeah, I mean, obviously from a Thermo Fisher perspective, I've had the pleasure of meeting many people in this room. We cover obviously on a next-gen sequencing side. We've got solutions that are built on the Sanger sequencing component. We've got pieces on the sensititer piece and on the culture component, and then on qPCR components as well. And so, as an organization, it's kind of interesting to see we kind of play that full gamut. And our learnings, and I think it goes to some of these comments of we had different organizations. Imagine in a big organization, there's a lot of and I should be careful since I'm being video recorded here, but there's some silos within big organizations, right? And so everybody's like, we can do this, right? And quite honestly, everybody on this panel has a technology that can do it. And the question really comes back down to the applicability, and I think it goes back to the last presenters, or the last question that came out is, how do you actually deploy it? Like, what's the impact? And I think there's kind of different fits and different needs. So for me personally, as I've seen this, is next-gen sequencing panels have been great and they offer a lot of value, but there are limitations, cost being one, workflow, even in core facilities. You have to have even core facilities. I think you go down a little bit further and you can say there are certain core facilities that have the right kind of skill sets. So you go down to the point of care component, or even the gold standards on culture. I mean, it is, whether we like it or not, it's, the technology is still the dominant technology that's out there in play. So how do you actually convince and help people get to a spot of being able to leverage some of these molecular tools which in the end is what we're trying to do, I think, within this group, hearing from, those that were brave enough to kind of share their story today, is like, that's the real world, right? How do we impact the drug resistance on these people's lives? What impact do we play as organizations to use our technologies of, yes, people are looking at us to use our technologies to make money, but how do we actually do it so that it's sustainable, scalable? So that leads me to my comment of looking at the centralized testing environments. I still think there is plenty of space that can be done, should be done on centralized testing, qPCR and Sanger sequencing, even though it's archaic technologies and comparatively, I think there is still a lot of value that's associated there in conjunction with many of these other technologies that we've talked about today. So.
Yeah, and just to take on, and I guess tomorrow Madhukar Pai will be presenting and talking a little bit about centralized, decentralized, and then patients, how that. So I think that they'll start getting into a little bit more of that, and I have another question to follow up with that, but I thought I'd give Christiana a chance to speak as well.
Okay. Yeah, thank you. So, I mean, you've seen the MySeq a lot in these presentations. It's certainly our workhorse for infectious disease detection. For us, TB, for me, it's dear to my heart because that's my background. But for us, it fits nicely into this global health concept, where we've been working with the FDA on the side of food outbreak monitoring a lot. All US centers for food outbreak monitoring have MySeq instruments, not the DX one, but the normal MySeqs. And maybe one point on the DX. So the DX instrument is a DX instrument and a normal research instrument. So actually, sites can do both research on that instrument, but there's a locked up component for the diagnostics. But coming back to this One Health concept, so together with the FDA, we've been building up, or the FDA has been building up a massive database for food outbreak monitoring. And I think for us, it's been the first very well accepted approach where we start to manage to control outbreaks before they happen. And they've shown wonderful data where we can get down to making these shorter and shorter until we react. And I think that is a very good example of what we are trying to do here in a context of infectious diseases. So there, the culture time is is faster, so we can do whole genome sequencing. We had a lot of discussions with Tim where to go with this. Should we enrich, should we do shotgun metagenomics, which is on Illumina's whole other end of the spectrum, where we are heavily involved in shotgun metagenomics. In fact, that's one of our biggest areas with the larger machines, and that's actually nice because we are extendable. If there are more samples, we do have the larger instruments. I mean, we just launched this gigantic instrument with the aim of getting the cost even lower, which is an overkill for this. But we have been discussing, and I think in the future there will be room for shotgun metagenomics. If we push this 10 years out with a really deep coverage, with really the accuracy which these instruments provide, and that has to come with databases, that has to come with this globalization. So I think we are on a good path here, and I hope we can peak at the small success stories which are out there already and drive it into the same direction.
Do you think that will help drive adoption outside of, let's say, North America or Europe, things like that, where lower and middle income countries, once you start tackling the cost and cost starts coming down, that's going to be another driver for countries to not only adopt it, but adopt it for additional purposes?
Yeah, and I think it was said before, it's a platform, and we can develop more and more workflows which feeds the same platform, which isn't potentially, which feeds the same run, right? Be it a food outbreak, a salmonella versus a TB, whatever. It's really a platform which can also monitor oncology assays, etc., and bring this technology into the low and middle income countries. And
especially as we're starting to see more and more oncological issues
for
happening
example,
in those
Africa,
countries.
yes.
So that would be great.
I mean, just to add on to that is the idea of, as we often, as industry folks, and in many cases we're like, well, our instruments, but it's really not about the instrumentation, it's about the answer, right? It's about being able to get the diagnosis, and there's multiple ways in which you can actually do that. But if you have to keep in consideration a business model, and it has to be scalable, right? If the idea is like, hey, we're just going to have some magical pot of money that's going to constantly fund it, it's probably not going to work. And so in the end, as you start thinking about these other technologies or these other solutions that, how do we engage not just a World Health Organization lab, but how do we engage like commercial reference labs? So I was talking with Chris over lunch, is to say, you think about in India, for instance, right? A lot of samples aren't processed through WHO labs. So how do you actually engage in those groups? And I'm sorry, I forgot your name already off the top. Jeff. Yeah. So we were talking about this earlier, is to say it's finding business models then that actually resonate, right?
Yeah.
And I think that's going to be a really key, important piece for adoption.
So
Marco,
on day 4.
I know you had one more question, but
No. I was going to say on
okay.
day 4, I was just going to put a plug in on day 4 that we'll have a discussion around that. But I know that we're running
a little
late.
over time, yeah.
Right,
Well, actually, we're right on time, aren't we?
Maybe my watch is fast, I'm sorry.
Maybe my watch is slow. But I do know that we have a reception at the ballroom on the first floor, so I invite everybody just to go up there at 7 o'clock, and there'll be plenty of food and drink.
Thank you all for the robust conversation today and for each of your presentations. Thank you very much.
Thank you.